Well, hello everyone, and welcome to our last talk on the eco-evolutionary dynamics and the making of the biodiversity symposium. I'm more than pleased to introduce Dr. Oskar Hagen. He's currently a postdoc in IDIV in Germany. He's a Brazilian like me, and he's also my personal friend for a long time. So I'm very happy to ask Dr. Oskar to join us and present his wonderful work. Thank you, Oskar, for accepting the invitation. Oskar will be taking questions in Portuguese, English, and Spanish. So thank you, Oskar, and welcome.
Hi, Manolo. Thank you very much. The pleasure is all mine. And thanks everybody for tuning in. Let me share my screen now. Hope it's working. Okay, so hi and welcome to my talk. I think we're set in. Just let me know if something's not working. Today we'll be talking about eco-evolutionary mechanistic models. Sorry, Manolo.
I was going to say that it looks good. The thing's just disappeared.
Perfect. Cool. And as Manolo already introduced, we'll be talking about eco-evolutionary mechanistic models, deep time, Paleo-reconstructions, and the origins of biodiversity, which is a little bit more of a methodological and a little bit of a different approach than the other talks in the session. And I think it's a very nice and adds up to this whole diversity perspective as well. My name is Oskar. I grew up in the countryside of Brazil, in Porto Feliz, where I studied informatics and conservation biology together with Manolo. And I studied environmental sciences, and now I'm doing the postdoc at IDIV. And the talk today would be mostly about the research I have been doing for the last 5 years. And first I will present the general motivation behind my work, including the research questions and objectives. Then I will explain a little bit about the biodiversity model I developed to tackle these questions. And then I would like to tackle some large-scale biodiversity patterns, such as the latitudinal diversity gradient, the Pantropical diversity disparity, and the code-adapted plants diversity. I will finish it off by briefly concluding and discussing some limitations and perspectives of this approach. Biodiversity is unevenly distributed around the globe. As you can see in this richness map of vascular plants, with some places with few and other places with many species. And this diversity has very characteristic patterns, such as the latitudinal diversity gradient, with an increase of diversity towards the equator. And I'm generally interested on why are there such distinct patterns, and also what processes shape or shaped biodiversity. We know that biodiversity patterns emerge from multiple processes, such as species interactions with biotic and abiotic conditions, speciation and extinction events, as well as species adaptations to past environmental conditions that change over time, such as continental drifts, mountain uplifts, and changes in temperature and aridity. And these processes, they interact in complex ways that have to be considered in order to move beyond statistical approaches. And this complexity is a strong limitation to our mechanistic understanding of biodiversity, but luckily this can be assisted by computers, since experiments at deep time and global scale are hard, if not impossible, to be conducted. So to assist the answering of my research questions, I developed Genesis, which stands for a general engine for eco-evolutionary simulations. And it's a framework that allows eco-evolutionary feedbacks at different time scales, considering dynamic landscapes. And it's implemented in R and C++, striking a combination of speed and code simplicity. so that users can implement their own eco-evolutionary rules or mechanisms just by programming in R. And Genesis allows the formalization of biodiversity theories, creation and testing of single or multiple hypotheses, and allows the historic simulations as well as future projection, helping us to quantify these processes that come from different disciplines and ultimately shape biodiversity. This thus enables spatial, temporal, and mechanistic exploration of biodiversity dynamics. So now let's jump into how this model works briefly, which would be very important for the rest of the talk. So the model consists of 3 parts, as you can see here. The inputs are dynamic landscapes, and the configuration files inform the initial conditions and the eco-evolutionary rules that play out in a specific model. And these are then fed into the main engine, which computes species abundances and distributions, populations divergences, phylogenies, and trait values, providing thus outputs that can be then compared to empirical data. So now I would like to show one example of such a landscape, so you can have an idea of what we are talking about here. This is a reconstruction for the entire globe for the last 200 million years ago that I compiled to input in Genesis. You can see on the left-hand side air surface temperature maps, and on the right-hand side aridity indexes. And this is changing through time and was compiled using plate tectonic models of movements and topographical changes, lithological information, stable isotope, and fossil data. And these dynamics landscapes in discrete time steps are fed into the main engine that has these four main parts, as you can see here. So to explain the model, I would use a very simple landscape here, composed with a highland, some lowlands in green, and a river, and a single brown fox species population. Connected populations exchange genes, and disconnected populations increase in divergency. So if they get into contact, they decrease divergence, but if they are isolated, they will gain divergence, and if a divergence threshold is reached, then speciation will happen. In this example here, there was no speciation event. Species then disperse in the landscape based on their dispersal ability and the landscape connectivity, which considers geographical barriers and distances. For example, such a barrier here would be this river here, for example. And after dispersing, these two fox populations suffer mutations, as you can see here, becoming the red and the white fox. And at the ecological module, unfit populations, they suffer local extinctions after abundances are adjusted. So in our example here, you see that the red fox mutation was unsuitable to the lowlands. However, the white fox is well adapted to the highlands and thus survives. And important to notice here is that all of these processes that I just mentioned, they can be modified over the configuration file. So for example, ecology can be made completely neutral, or take multiple ecological rules, such as the inclusion of more complex competitive interactions and environmental carrying capacities, for example. Speciation here can account for interactions with abiotic conditions, such as the effects of temperature towards speciation rates. And dispersal, for example, ranges or can range between different species related to specific traits and can be made deterministic or completely stochastic based on a probability density function. And trait evolution can be random or dependent on species or environmental conditions. So now that you got an idea of the input that we use and how the model works, let me show you an example of an output. So here there is an example of one simulation, so showing species richness emerging over time. So we start here with one single ancestor species spread out to the entire globe. you can see here in the blue, around 65 million years ago, mainly after the mass extinction of the Cretaceous Paleogene. And this generates then latitudinal as well as longitudinal diversity gradients. And you see the emergence of this diversity. You would see also the flashing around 2.5 million years, there you go, which are reflecting actually also climatic oscillations from the input data. So such outputs, for example, can then be compared to empirical data, and we can use summary statistics, for example, here the LDG curves to match observed to empirical data. The power of such a tool or such tools are that it allows the comparison of multiple mechanisms and landscapes within a common framework. So in this example here, we use two different reconstructions. Sorry, I need to minimize this. We also use different configuration files. So you see these 5 different configuration files representing different hypotheses or different eco-evolutionary rules. And this then result in this all different outputs. So for example, we have LDG curves and slopes, we have alpha richness, we have species distribution ranges, we have also phylogenetic balances containing evolutionary history informations, and also lineages through time plot data. And in this example here, while we were investigating specifically the LDG, we found that considering energetic carrying capacity simultaneously produced multiple biodiversity patterns. So was one of the models that could reproduce the biggest amount of multiple emerging patterns contrasted to empirical evidence, suggesting the importance of these ecological limits when assuming niche conservatism. And our simulations show that simulations having a high speciation rate and a low dispersal ability tended to lead to higher or stronger latitudinal diversity gradients. Sorry. Okay, so now let's go towards the next large-scale pattern, which is the Pantropical diversity disparity, or the PDD. And this derives basically from tropical moist forests that observed are the most species-rich terrestrial biomes on the planet. And it's been documented for many different groups that Afrotropics has a much lower species richness compared to the Neotropics and the Indo-Malayan tropics. So for example, here you see the numbers for palms, but studies showed a consistency throughout different groups as well. In making Africa even being called the odd one out because of this richness contrast in tropical moist forests. And there have been many different suggested hypotheses to these contrasts, mainly deriving from current versus past environmental conditions, lower speciation versus a higher extinction in the Afrotropics compared to these other regions. So in order to tackle this, we first compiled empirical data for plants and vertebrates, of which 72 and 81% showed this PDD pattern, meaning a lower Afrotropical diversity. And you can see here in these ternary plots, distribution of plant groups and vertebrate groups. So each point here is a plant group, and you see that if there are equally balanced, they would be more or less in the middle, or if they're more present in the Neotropics, they will be more concentrated here. And here you see one example of palms, and this it's alpha diversity richness. And here you see for vertebrates the trogoniform birds with a higher diversity in the Neotropics. We then did some simulations starting at 110 million years ago, where the continents were mainly still taking the shape of the current distribution. And we started with one ancestor spread out throughout the globe at the terrestrial ecosystem. We then modeled different niche weeds, different dispersal shapes, and multiple divergence times, considering an evolving niche optima for temperature and excluding arid sites. And these are the results of the simulations. So you see here, each of these single points is one simulation. And here we select one simulation and we plot the alpha diversity or the richness map. But not only, as I showed before, the idea and the advantage of this is that we can look at multiple patterns. So besides just looking at spatial diversity, we look then at the community relatedness index, mainly the net relatedness index, NRI, where we look at communities, local communities, and the clustering of species. So if species in a community are highly clustered, they will have a high NRI, as you see here, these two red species. And if species are a community composed of two species less clustered, they would have a lower NRI. And these are NRI maps and the phylogenies for palms, Anura amphibians, and best matching simulation. So simulations show that multiple different processes acting in concert best explain this richness contrast, while considering past climatic changes was superior than only looking at current climate. And these best matching simulations, they provide insights on this diversity dynamics, showing that the Afrotropics lower richness was driven by both a lower speciation and a higher extinction compared to these other regions. And this is driven or probably driven by a more extreme cooling and aridification, which much less historical environmental heterogeneity of the African continent. And this can be related to the fact that the African continent lays in the middle of a tectonic plate with much less tectonic activity. To finally confirm these findings, what we did was then to modify these reconstructed landscapes we used. So here, for example, we removed the Andes. So we flattened the Andes out and simulated again, and we got the expected resulting curves. So a much lower richness when we remove the Andes than when we have the Andes in it. And we did the same for the Indo-Malayan tropics regarding heterogeneity and also connectivity between islands. And also by removing the aridity constraints of the African continent, we see that we get consistently higher African richness. So now closing the hot and tropics, we move to the cold and cold adapted diversity, where I looked into the life history of cold adapted plant species. And we know that mid-latitude mountain ranges are richer than Arctic areas. And there have been many suggested hypotheses for that. And one of these is that the Arctic was colonized by lineages that originated in mid-latitude mountain ranges and then migrated. In order to test this hypothesis, first I did a synthesis of the location and time of this cold-adapted plant origins and combined this with Paleo-reconstructions of temperature for the northern hemisphere. So here you see the minimum temperature of the northern hemisphere and these multiple different regions. And you see basically that these names here are cold-adapted plant genus, and based on the literature, more or less the arriving or the first ancestor time, and they're dashed or they're dating back to also the location of occurrence or more likely location of occurrences. And we see that most of this cold-adapted genus originated in Himalayas, especially after this temperature drops triggered by the uplift of the Himalayas and later the cooling of the Arctic. So you can see from our paleo-reconstructions through time that the Cenozoic was predominantly warm, and temperature decreased in the late Oligocene, Miocene, right here in this arrow, by the collision of the Indian with the Asian plates, giving this, providing this uplift of the Himalayas and first opportunity for the appearance of cold-adapted lineages. In order to grasp this occurrence and adaptation of cold-adapted plant species towards this new niche, we started multiple simulations at multiple times, maximum at 55 million years ago. Before that, we assumed, okay, the world is warm and we wouldn't expect to see any cold temperatures. I just hear a to-do, but I hope it's everything fine. And we used one or multiple ancestor species, and we used also multiple temperature optima and competitive abilities, which evolved and traded off. Here you can see this reconstruction of a standardized richness of cold-adapted floras through time, based on this fossil calibrated phylogenies. These are the simulated appearance. So we see a very good match temporally, but also we could get a very good match spatially, showing that these cold-adapted floras most likely emerged in the Himalayas and later colonized other regions rather than originating in these other regions independently. This provides insights on the origination and maintenance of cold-adapted plants, which can be used to forecast or to gain knowledge or gain expectations on what will happen under global warming projections. So these are preliminary results, but I use this as an example how we can use this kind of information to see where we will have a strong loss of species. So we see that this dispersal and adaptive abilities that allow cold-adapted plants to track climate dynamics in the past would not be sufficient to prevent large cold-adapted species loss, especially in mid-latitude mountain ranges. So concluding, limitations are many, specifically regarding paleo-environmental reconstructions uncertainties, but also biodiversity data, especially past information. And also model complexity and computational time is an issue as we gain and increase resolution of paleo data, or we go towards a more lower resolution in levels of organizations, which relates with perspectives which are as many as the limitations, or maybe even more, which would be to include additional mechanisms and paleo reconstructions that can, earth science can feed on life science data and information. So you can use biodiversity data to inform on geological reconstructions or vice versa. And next steps and perspectives, and this relates a little bit more with the work I'm starting to do here in Germany, which is to integrate biodiversity across different levels of complexity, time, and space. And also we can use such spatially explicit eco-evolutionary models to model alternative complex systems that could use information from biodiversity and evolution theories, such as human language, belief systems, or even technologies. So computer models are promising tools for investigating multiple mechanisms proposed to result on different biodiversity patterns. And this hopefully would catalyze interdisciplinary research and advance our numerical knowledge on biodiversity towards the unification of biodiversity theory. And with that, I would like to conclude with phrase from Kirchner and Gerhardt, which I like very much, and I think reflects a little bit the research I do, which is understanding life is not a conquest, but a slow lesson in appreciation. And with that, I would like to thank you for your attention. I would also like to thank my former group and my former supervisor, also many different collaborators and people working with this, especially SBE committee and Manolo for inviting me here as well and giving me this opportunity, and for creating this platform where, I don't know, language and income is not a barrier for science. And I put myself to questions. You feel free to contact me over Twitter or email. If you'd like the package, feel free to try it out. It's available on Cram. And that's it. I'm happy to take questions.
Thank you very much, Oskar, on this very exciting topic. I personally like it very much. I find it fascinating. And we have time for questions. We already have some in the chat. I will read it. So the first one is in English, and I'm sorry if I pronounced the name incorrectly, but it's from Moomin Michael Menting. And he asks, does Genesis requires large data sets to be analyzed? And what are the minimum requirements for the data sets to be included in the analysis?
Okay, so it doesn't require large data sets to be analyzed. So we did at the beginning kind of sandbox experiments, and a little bit of my postdoc now would be also using Genesis or expanding Genesis to purely theoretical experiments. So here we basically create some island systems or some sort of dynamic landscapes, and we see how evolution being integrated in a meta community theory would result in different patterns. And of course, it depends a lot on the question that you are asking. So if you're investigating, for example, evolution of certain groups of plants, of course, fossil data would be necessary. But the idea and the perspective here and how I started this project was mainly to say, well, we don't know enough about the paleo environment, we don't know enough about the mechanisms, but we have a lot of verbal theories that are not formalized, and can we at least create something where we bring all of this data and theory in a same place that is formalized, and we start discussing and testing these things. So you can even use something completely free out of data where you say, well, how can we disentangle these two competing theories? You simulate things, and maybe you see signatures in the resulting and emerging patterns that you can then use to search for empirical data. I don't know if I answered all of the points, but the minimum requirements, and I think computational, of course, most of the time you need several runs, so you can do a little bit locally, and I use my machines for some runs, but most of the time what you would like to have is a cluster computer where you can launch a lot of simulations and analyze the behavior of multiple parameters. But it's also highly dependent on the number of parameters you choose.
Perfect. We have another question, this one in Portuguese, so I'll switch a little bit. Pergunta da Genilda Castro de Almeida Neta. Parabéns pelo trabalho. E existe perspectiva de novas implementações do pacote esse ano?
Bom, Genilda, obrigado. Existem sim perspectivas. Eu acho que a maior parte delas são resolver alguns bugs que tem e conseguir consertar alguns features, não essential bugs, mas, por exemplo, a função de reiniciar as simulações que estavam existentes, mas elas acabaram saindo do pacote no final por dificuldades de implementar no RCPP e outros solvers em C++. E aí a gente acabou tirando isso, mas a ideia é voltar isso sim para lá. E o pacote está disponível também no GitHub, então a ideia é fazer GPL3, então a ideia é deixar disponível para a comunidade científica. Então, tem issues, se já tivemos também pessoas escrevendo, o scale não é ideal para colorblind people, então já tem aquela issue, a gente quer resolver isso também, e a gente vai chamar pessoas da comunidade, né, que quiserem programar ou quiserem usar para um uso específico, e vamos ver, tomara que sirva para mais pessoas e acaba gerando, assim, mas por enquanto eu estou me recuperando do meu doutorado ainda.
Legal, legal. Ela já agradeceu aqui a resposta e desejou sucesso aí nas próximas etapas.
Obrigada.
A próxima pergunta é em inglês. It's from Natalia Caldeira. She's a Brazilian as well, so you can answer in Portuguese if you want. I will read the question in English just to make sure I read the precise question. Oskar, thank you so much for the presentation. I'm wondering whether Genesis takes the differences in preservation data into consideration when handling fossil information, and whether you have tested data with different preservation rates to understand whether your method has a limit, hard or not, for its applicability in that regard.
That's a very cool question. And this is one aspect that I thought of when I did the implementation of Genesis. So basically, when you have all of these processes, so the idea behind is, okay, it's a virtual world and we understand completely. This is a model, so we know what's going on there. And we coded the rules. But of course, we want also to allow for the observational bias. So there is a part, which is the observer function, that you can code and build in kind of the observer degradation of the data. And there you could include a lot of different functions. So you can say, well, let's consider preservation rate at different time periods as being different, or at different locations, or being higher for organisms with a bigger body mass if you model body mass, or dependent on abundance if you model abundances and not just occurrences. And this is actually, there's two projects going on right now which are using Genesis and are using multiple different observer functions to kind of validate methods to reconstruct real data. So if you have, I don't know, methods of getting the speciation rate, or you have methods of reconstructing any aspect of life, you basically can say, okay, let's use a more realistic model where you say it's a spatial and we understand the rules, and we see if we can, by degrading these or by different levels of degradation, we still can gain information of the real processes behind. So it's more like a tool to, you could use it to improve reconstruction methods and also to simulate close to real data.
Awesome. We have another one in English as well from Neander Hemming. Congratulations for the fantastic work. What is the temporal resolution of recent climate datasets?
Okay, this is a cool question. I mean, I'm a biologist by training, but I had to jump into this at the beginning and compile this kind of data sets, and it depends a lot on the on how deep on time you go. So basically, all of the examples I showed in the presentation, they started, the resolution was 170,000 years. So one time step is 170,000 years, which is a lot. But let's say there are some studies. We did this at the very beginning of just the core of Genesis. We used this to test for the last, I think we're 21,000 years, and we just used at the reindeer caribou populations, and we just looked at genetic data, and there we have much more a higher resolution. So it depends a lot on which kind of input you're using and which kind of research question you're using. And it's fair to say that the deeper that you go, the less resolution, spatial and temporal resolution you have. And for this, I decided to use to degrade the higher resolution that we have towards the present, but keep it more or less constant, because we don't know how temperatures oscillated in the deep past. So for example, at 40 million years ago, we have a resolution of 1 million year, or this 170,000, which are interpolations. But there it was kind of a compromise to say, okay, let's limit it at least close to what we know the best, and future prospects are huge. I mean, for example, using circulation models or climate circulation models to kind of predict seasonality or more fine fluctuations in climate, and then simulate this. So this is like really a big step. But as far as I know, if you go deeper than 50 million years, yeah, if you over a higher resolution than 1 million year, or yeah, couple 100,000 million years, 1,000 years is not really realistic.
Great. Next question is in Portuguese, e é uma pergunta da nossa amiga professora Isabel Bonatelli.
Olha lá.
Brilhante palestra, gostaria de saber como as espécies invasoras estão sendo pensadas no modelo.
Eu tive uma conversa antes de ontem numa reunião aqui, porque no grupo que eu tô, um dos grupos que eu tô colaborando trabalha muito com espécies invasoras, né? E aí vem muito da complexidade, né? Do padrão atual. E eu tive essa ideia, né, nessa festa, nessa discussão sobre o uso, por exemplo, do Gênesis nessas espécies invasoras, que seria provavelmente modelar espécies sem uma influência humana, ou sem uma dispersão forçada, né? E com isso você mais ou menos consegue ter comunidades específicas baseadas no conhecimento, nos modelos que a gente acha que são corretos. Então, sei lá, essas espécies evoluíram e estão adaptadas a esse tipo de oscilações climáticas ou esses tipos de comunidades. E depois, baseado nessas simulações, simplesmente trocar espécies ou introduzir espécies experimentalmente no modelo e ver o que, será que a gente consegue concluir ou consegue tirar informação disso. Agora, espécies invasoras, assim, no sentido da influência humana na dispersão disso é muito difícil, né? Porque a gente está... Esse é um modelo mais, assim, de origem de biodiversidade em deep time. Então, isso, a maior parte das vezes, quando a gente pensa em espécies invasoras, eles são um ruído ou um noise no data para comparar as simulações, mas poderia-se usar isso para tentar entender o quanto a gente sabe, o quanto do histórico evolutivo dessas espécies invasoras teria uma influência a longo prazo, talvez até.
Maravilha.
Pena que eu
Muito
não
obrigado.
estou vendo a cara das pessoas. Eu queria ver
a Isabel e a Natália responderam agradeceram as respostas. Eu queria agradecer também de novo imensamente a sua participação. Muito bom. O Neander também aqui respondeu, falou que também it's a huge effort to compile all these datasets. Verdade, indeed. And eu queria agradecer um monte, convidar a todos agora, que a gente tem uma sessão de pôster agora no Discord. Oskar, I would like to thank you so much for joining us, for accepting the invitation. Now we have a poster session in Discord, so everyone, if you can join us there, and thank you very much, Oskar.
Manolo, thanks so much for the opportunity. I mean, it's all my pleasure, and I'm really glad to be here and to get a lot of questions and interesting comments and regards, and for this opportunity. And thanks for organizing this. I mean, it's a huge effort, and appreciate it.
Thank you. I'm just one single person in this organizing committee, which is amazing. So it's a important effort indeed. Thank you very much.
Totally. Thanks. Cheers.